Okay, so plan is to talk to you a little bit about the next topic on essentially part two of photosynthesis, and then toward the end, second half, I should say, the second half of the lecture, I'll go over the exam stuff and have time for you to ask questions. So the exam's this Friday. But let's first talk a little bit about, I guess the fancy way to talk about it is called carbon fixation, which is now the idea that the plant will take in carbon dioxide and use that to make sugars. And so in order to do that, they have to have enough reducing molecules and they have to have enough ATP in order to get that to happen. So that's all happening through the light reactions, which we've already covered. And this idea that, you're peering into the chloroplast and you are looking at the thylakoid membrane that's floating around inside the chloroplast. And so by the time the light reactions are done, you have that lovely NADPH and the ATP, and that's all being produced in the stroma, which is really good because that's where the carbon fixation will start to kick off. And so once they, excuse me, once they have enough NADPH and ATP, they can then feed that in to parts of the reaction that make sugars. And so that's the next stage. So here you're seeing the link between the two phases of photosynthesis. You have the light reactions on the left here. They're trying to show the ultimate donor of water, stripping electrons off of that, feeding it into the electron transport chain process, and then NADP plus is the ultimate acceptor. And so then you get the NADPH and ATP, and then they're trying to show how that immediately is going to get used right in the stroma for the carbon fixation. So the carbon fixation, taking in that CO2 through the stomata, and then making carbohydrate. It's often referred to as the Calvin-Benson cycle. I tend to just shorten it down to the Calvin cycle. It also has various other names, carbon fixation, of course, Calvin cycle. Some people call it the C3 cycle. So you might hear me I tend to say Calvin or C3 cycle, but basically it's the idea of now we finally think about what's happening to that carbon dioxide that the plant is taking in. It's also called the photosynthetic carbon reduction cycle, which is a mouthful, and that abbreviates down to PCR. I don't know about you, but when I hear PCR, I think polymerase chain reaction. I do not think about this long phrase, so I don't use that one at all. But just be aware that sometimes you'll see that in the book or the literature. Okay, so Calvin cycle or C3 cycle, named after Dr. Calvin, who kind of was pivotal in getting the final difficulties of the Calvin cycle ironed out. So they call it the C3 cycle because the first molecule that they could identify in the cycle is 3 carbons. And so they started calling it the C3 cycle. We definitely see that in certain phases, you got to use ATP and NADPH. But they were really struggling with what does the carbon dioxide react with? So the plant brings it in, and if it's a 3 carbon sugar that is made, that means that it's probably a 2 carbon molecule because 2 plus the 1 carbon in CO2, that's 3. So they were hunting around for a 2 carbon molecule that CO2 could react with and couldn't find anything. They were scratching their heads like, can't find it. Then Dr. Calvin came along and said, well, there's more than one way to get to 3. What if you have CO2 react with a 5 carbon? That brings it to 6, but then if you split it in 2, then you're down to 3. And that's exactly what they ended up finding. It was a 5 carbon sugar that reacts with CO2. And then what happens is very quickly it breaks in half essentially, and you get two identical molecules. So that 6-carbon molecule that first gets formed is so unstable and doesn't last very long. It quickly breaks into 2 3-carbon compounds, and they kept missing it. They didn't have the sensitivity of the test to actually see that 6-carbon. So that's why they missed it. And Dr. Calvin came along and for his efforts won the Nobel Prize for figuring this out. He actually went to Michigan Tech University and did his undergraduate degree there. His full name is Dr. Melvin Kelvin. And he is famous for helping the discovery of the Calvin cycle, C3 cycle. Okay, 5-carbon sugar. Ribulose 1,5-bisphosphate, which thankfully we can abbreviate down to RUBP. So 1,5-bisphosphate, that means there's 2 phosphate molecules on there. And this is this is the full Calvin cycle, but what we need to zoom in on is the CO2 coming in and then the ribulose bisphosphate. And when you get that reaction, you eventually end up with phosphoglycerate, which is 3 carbons. Okay. So let me see if I can find the. So this is the full reaction that they missed initially. They didn't see this unstable 6-carbon here. But basically, here's your ribulose 1,5-bisphosphate on the left, CO2 coming in, they show where it connects. And then that 6-carbon quickly breaks into 2 identical molecules of PGA, phosphoglycerate. So phosphoglycerate, another way to say that is phosphoglyceric acid. So that's why it abbreviates down to PGA, phosphoglyceric acid, or phosphoglycerate. Either way is fine. So you can abbreviate that. You don't have to write that all out on a quiz or exam. You can abbreviate that down. So what we see happening is RUBP, that 5-carbon sugar, gets carboxylated. That means CO2 is added to it. You get that unstable 6-carbon, and then that breaks down into 2 identical molecules of 3-PGA, phosphoglyceric acid. So 3 means that there is a phosphate on the 3rd carbon. That's what 3 is referring to. So the enzyme that facilitates this reaction of taking the CO2 and reacting it with RUBP is called Rubisco. And I do need you to know that. So Rubisco, that's an acronym. It stands for ribulose 1,5-bisphosphate carboxylase oxygenase. So thankfully, it comes down to this nice handy acronym of Rubisco. But I wanted to spell it all out because it's important that you see this weirdness going on. There's a carboxylase word in here and an oxygenase word. ASE on the end of a word in science means it's an enzyme. So what this is implying is that this is a dual function enzyme. It does 2 different things depending on the state of the cell and what's going on. It can carboxylate, it can be a carboxylase type enzyme, or it can be an oxygenase type enzyme, which just means carboxylase means if it's doing that function, it's taking in CO2 into its active site. However, if it's running its oxygenase function, then it takes in oxygen into its active site. And for the Calvin cycle, that's not what you want. You want CO2 coming in to the active site of the enzyme. So Rubisco is often referred to as a sloppy enzyme. It's sloppy because it can take carbon dioxide in or oxygen, and it just depends what is more abundant around it. So it's a competitive reaction. Whatever is more abundant, that's what comes into the active site. we'll talk more about that. That has some serious implications for the plant to deal with. This is the most abundant protein in the world. All organisms that do photosynthesis and carbon fixation, well, nearly all, almost all, have the Rubisco enzyme. And so when you look at just the leaf tissue, about 40% of the protein content is Rubisco. So super important enzyme. If you don't have this functioning correctly, you cannot complete photosynthesis and get your sugars made. Okay, so Rubisco is facilitating this first step right here, this carboxylation. So you have to imagine Rubisco sitting there, taking in CO2, taking in RUBP, and reacting them together to ultimately get you these two identical molecules of PGA. So that's the kickoff. Now, when you look at it, so we're back to this. I call this the pie chart, by the way. So if I talk about a pie chart to on a quiz or exam, this is what I'm referring to. So when you look at this, what we just described is being shown right here on this part of the pie chart. Do you see ATP or NADPH? No. So that means this will just happen spontaneously. You don't need to push it with a reducing molecule or ATP. If Rubisco sees CO2 and RUBP, it's just going to happen. It's exergonic, it's just going to go. So that's good. Then if you look at the phase 2 and 3 of the pie chart here, now you start to see the NADPH and ATP playing a role. But the first step is all good. It doesn't need ATP or NADPH. So we call it carboxylation because RUBP is getting carboxylated, which just means you're adding a CO2 to that RUBP. So the first step is carboxylation. That just happens all good if Rubisco is seeing the carbon dioxide and the RUBP around it. The next stage is called the reduction phase. Reduction. So this is referring to the idea that NADPH is coming into play. Remember now, NADPH is called a reducing molecule. Reducing molecule. That means it's very happy, chemically speaking, to give off electrons into a reaction. And that's what we're going to see here. You also see that ATP is involved. Okay, so with the reduction, we start with the PGA. And there's an intermediate compound, which you don't need to know that. And you end up with glyceraldehyde 3-phosphate. So you go from PGA to G3P. You can abbreviate down glyceraldehyde 3-phosphate to G3P. So G3P, this is another 3-carbon molecule. They often will say it's a triose phosphate. Triose just means it's 3 carbons. I'll show you that in a little bit more detail. So what happens is, if this reaction, the first part of the Calvin cycle, the carboxylation, and then the reduction, if it's all cooking along nicely, you're going to get a buildup of G3P, and some of it can get sent out of the chloroplast and into the cytoplasm. And if that happens, it gets converted just through a rearrangement reaction into dihydroxyacetone phosphate, which is abbreviated down to DHAP. So we'll look at that later, but you're going to see that it can devote some of the G3P that's building up, get it out into the cytoplasm. And actually, that's where you're going to start to see the actual sugar getting made, is in the cytoplasm. But most of the G3P that gets made will continue on in the Calvin cycle, stay in the chloroplast, in the stroma of the chloroplast, and it's necessary to finish off the Calvin cycle. Okay, so the reduction phase is, if you can remember that NADPH is a reducing molecule, so that's why they named it the reduction phase. So here you see PGA on the left, and you can see it gets phosphorylated. and then it gets reduced by NADPH into G3P. So this last molecule is G3P, glyceraldehyde 3-phosphate. So it's still a 3-carbon. All right. Okay. So in order for reduction to happen, you got to use ATP and the NADPH, and that's coming from the light reactions. Okay. Then the last stage is regeneration. That word is being used to refer to the idea that you're going to regenerate RUBP. And so you see the glyceraldehyde 3-phosphate as the starting point of this last phase. And you go from glyceraldehyde 3-phosphate back to a 5-carbon. So remember, RUBP is 5 carbons. So you have 3 carbons and you have to get to 5 carbons in order to get this to happen. So this pie chart makes it look like maybe they're fairly equal in complexity, but it's actually the regeneration phase that is most complex in terms of the amount of reactions going on there. So we're gonna need to work through that a little bit. But basically, when you think about it, the Calvin cycle has to be completed. You have to get back to RUBP so that the plant all day long has its, hopefully if everything's going okay, stomata are open, CO2's coming in. It has to have enough RUBP to react with, or everything starts to fall apart. So, but just be aware that people often call the RUBP the acceptor molecule of the Calvin cycle. That just means that it's accepting and reacting with carbon dioxide. So they refer to it that way. But basically, you got to have enough RUBP made up and ready to go so that as the CO2 is coming in all day long, it can react with it. So when you look at this last phase, it's a series of reactions. There's carbons that are or molecules that are 4, 5, 6, and 7 carbons long. And basically, if you look at it in a nutshell, what they're doing in this part of the reaction is that you have carbon from 5 of every 6 G3P molecules is being used to make 3 molecules of RUBP. So basically what that's saying is most of the G3P needs to get recycled and regenerated so that you can get the RUBP up and running. So I'm going to talk a little bit more about these 4, 5, 6, and 7 reactions. We're going to kind of take a big picture look at it. We're not going to get into the nitty gritty. But basically what we see happening here is that when you have that G3P, some of it will get exported, a minor amount will get exported out into the cytoplasm. And we'll talk about that later. Most of it gets put back into the cycle to make more RUBP. So this is one way of representing the regenerate. Well, actually the whole thing's on here. The blue up top is carboxylation, and then the reduction in green, and then this big pinkish peachy color, that's all the regeneration. This is nice if you like to count carbons and look at things. I find it hard for my brain to connect with. So I put this in here. If some of your brains work well with this, go for it. This is the one I prefer. It's a little more streamlined and just simpler to look at. This is again the full complete Calvin cycle, but the lion's share of what you're seeing here is regeneration. So what we have here is the start, RUBP and CO2. Now, for ease of counting carbons and doing all of that, we tend to teach it as, okay, let's think about 3 carbon dioxide coming in and reacting with 3 RUBP. Then the math works out nice and simple. So let's do that. So that's why there's a 3 there. So 3 CO2 and 3 RUBP react. So that's where Rubisco is involved. And then you get the PGA, which then gets reduced by NADPH, gets phosphorylated. And then you end up with this G3P. So if you have 3 CO2, 3 RUBP, you'll end up with 6 molecules of G3P. So they often refer to that as the G3P pool. You have 6 of them here. And this last one that they show, where it says DHAP export, that's the one that's going out into the cytoplasm. All the rest is being used to make more RUBP. Okay. So we're not gonna get into the nitty gritty, but there's a few things on here to key in on. I like to call it the numbers game, where we're counting carbons and the magical number is 5. We want to get to 5 because that's what RUBP is. It's a 5 carbon sugar. Where did it go? Okay, that's weird. All right, I'm going to stop share for a sec and pull up another document, another PowerPoint. Okay, I just, there's another PowerPoint in EDUCAT called the numbers game, C3 cycle, regeneration, phase, numbers game. That one I wanted to show you. I thought it was in this slide set, but it's not. Okay, so I'm going to reshare. Okay, let's go, let's look at it this way. Okay. So this G3P pool. Okay, come on now. Ignoring me. All right, so you have this G3P pool. All right, so those are 3 carbons. So what you see, let me go to this side. So those 3 carbons, those 3C, each of those represents a G3P. So if you react 2 G3Ps together, you get, if you add them up, it's 6, doing simple math, okay? And then if you add another 3 carbon compound to that, now you're up to 9. So let's go back. So here are 2 3 carbons. Oh no, here, let's do these 2. These 2, so a G3P and a G3P react and you get a 6 carbon. You don't need to know the name of it. And then what they're trying to show here is that another 3 carbon joins in on that. Okay, so if you have this 6 carbon right here, and then another G3P comes and reacts with it, now you're up to 9 carbons. They don't even show you what that 9 carbon molecule is. They just show that it quickly separates into a 4 carbon and a 5 carbon. All right, so that's what we're looking at with this idea here, that 9 carbon will quickly separate into a 4 carbon and a 5 carbon. 5 carbon's what we're going for. So we managed to get a 5 carbon out of that. So that's why I circled it. It's like, okay, now we got a 5 carbon. So let's keep that on the back burner here. Let's look at that 4 carbon. So now we have a 4 carbon. We got to get to 5 somehow. So that 4 carbon will react with another G3P. and become a 7-carbon. And then if you add on another G3P, then you're up to 10. And if you split those in two, you're at 5. So you've made the magical number of 5. That's what we're going for through that process. So let's go back. So that 5-carbon is here. That's good. We'll hold off on that. But it's this 4-carbon that we need to think about. So the 4-carbon reacts with another G3P and you get your 7-carbon. Another G3P comes in and you get your 10-carbon. And then that breaks into 2 5-carbons. So 5-carbon is what we're going for. Now they're not all RUBP yet. So this one is a xylulose 5-phosphate. This one's a xylulose 5-phosphate. This is a ribose, but they're close. They're 5 carbons. So what you see is this convergence point here where you get rearrangements going on that allow for ribulose 5-phosphate. Okay, so now you're up to 3 of those. And then you can see at this very last stage, you got to put in ATP because ribulose 5-phosphate needs 1 more phosphate in order to become RUBP. Because RUBP has 2 phosphates, all right? So you have to tack on another phosphate to that. So all of these 5 carbons, it's enough to know that they rearrange, they go through some reactions to rearrange to become ribulose 5-phosphate. But that's not quite there yet. You have to add on ATP now. And so ATP reacts with that, and then you're finally at the, the acceptor molecule, RUBP, the 5-carbon molecule with 2 phosphates. And now it's good to go. Now RUBP can react with more CO2 coming in. Okay, so as far as the regeneration reaction, okay, so what we just described is pretty much all of this. So all of that is represented by this. And this is what you need to know. You don't need to know all the names of the compounds or the enzymes that facilitate it. Know the numbers game. And that's good enough. This is not a biochem class. So we'll stick with the bigger picture idea. Okay? So don't get caught in the minutia of all the names and enzymes here. It's going for this, the numbers game. Okay. So we just went through the Calvin cycle. I think it's helpful to do that pie chart process as you're studying to chunk it out into those 3 different phases, because that's how I'll be talking about it on the exam, looking at the 3 different phases and what's happening in each one. Questions on, let me go back. Not that one. All right, let me go back here. Oh boy. Back to here. This computer. Yeah, it's frozen. All right, I'm gonna stop share. My laptop and Zoom just don't like each other. Okay, so we just went through this, the pie chart of the Kelvin cycle or the C3 cycle. So the whole goal is to get that REBP built up. And so that CO2, as it keeps coming in through open stomata all day long, there's enough REBP for the CO2 to react with. Because if you can keep that going, what's happening is this cycle keeps going around and around and around and around. And as that's happening, you get some of this G3P getting sent off into the cytoplasm where you can make sucrose. That's one huge goal, is that some of that G3P will get sent for sucrose production, and that's happening out in the cytoplasm of the cell. There's also, too, the idea that the plant could decide to make starch. So if there's enough sucrose circulating in its body and all parts of its body has enough sucrose and everything's good, then the leaf might say, okay, I'm going to store some sucrose for later use in case I run into an issue. So if they're going to do that, they don't store it. Most plants don't store it as sucrose, they store it as starch. So they'll build starch. And if that happens, it just stays in the chloroplast and you'll start to see starch building up. So it's kind of an either or thing. The plant's trying to figure out how much sucrose do I need to make? Where do I need to send it? And then if there's everything going just great, it'll decide, so to speak, oh, I'll make some starch. There's enough sucrose out there. I'll make some starch just in case we run into issues. And so it's kind of a, the Kelvin cycle is kind of linked into sucrose synthesis, but you definitely, as you follow what's going on with the Kelvin cycle, if that keeps going round and round and round, you definitely can have some G3P getting used for sucrose or starch. Any questions on the Kelvin cycle right now? One caution I have is that you please get straight which is the 5-carbon compound that reacts with CO2 and which is the enzyme. Please get that nailed down now. I've seen on the exam that students will mix those up. So RUBP is the 5-carbon sugar. Rubisco is the enzyme. And the way to remember that is that the Rubisco, CO, if you can remember that the CO stands for carboxylase oxygenase. ASE on the end of a word, that is the enzyme. Okay, so Rubisco is the enzyme that facilitates that first reaction. RUBP is the 5-carbon sugar that reacts with CO2. So please get that separate in your head so that they don't get mixed up. Okay, I will stop there with that material. So this will not be on the exam coming up on Friday, but I did want to save some time here for talking about the exam content. So let me pull up. Starts with the phloem translocation. And then mineral nutrition. Biological nitrogen fixation. And then the light reactions partial, not the actual step-by-step, but some of the intro material. Okay, so I pulled all those up. Let's go back to phloem. So phloem translocation, let me share this. And this. Okay. So phloem translocation, we're looking at once the plant is done doing the Calvin cycle and you finally get some sucrose that can be sent to other parts of the plant body. Now we're talking about phloem translocation. So knowing where it takes place, you know, vascular tissue, what parts of the vascular tissue, and you know, what are some of the major components. We looked at certainly sucrose is the number one, but that there can be some other things. It's not just sucrose. We looked at why they think sucrose is the molecule that's actually transported when there's lots of other options out there for what kind of sugar could get transported. So the idea that you have sucrose is small, it's a simple disaccharide, get a little bit of energy when you split that bond between the glucose and fructose. Okay, so some of the more simple things, you know, those are good for multiple choice. If you think about what could be an essay, you know, typically essay questions in this class would be like a process, you know, like where you have to describe something from start to finish and describe, you know, how it happens. So the process of translocation, you think about it, that's kind of hard to sometimes. It's a little challenging to make multiple choice for that to make it adequate for assessment. So this idea of translocation, where you have the movement of sugar from source to sink. So that's kind of a big picture idea because there can be, you know, different sources, different tissues that can be sources, different tissue that can be sinks when you think about the full plant body. So knowing what, you know, the overall definition of a source versus a sink is important. What about the translocation process? What kinds of questions do we have on that one? So we went over that process in lecture. You had a lab on it where you did the two different sugars in the dialysis bags and looked at the movement from source to sink. You do have to think about water potential in order to describe what's going on, because that's impacting how water will move. And so when you think about this idea of movement, it's a pressure gradient from higher pressure in the source and a lower pressure in the sink. Are you good on this, or you want me to work through it a little bit? Yes, no? Okay. So we have this idea of thinking about the sugars getting loaded from the leaf cells that are doing photosynthesis. They're going to start dumping their sucrose into the sieve tube element. So that's where we start to see this pressure building up. So it's right in the sieve tube element companion cell complex where you're going to get lots of sugar. Well, that's solutes. So you're dumping a bunch of solutes in there. And so more solutes mean a more negative psi sub S. Okay. More you load in there, your psi sub S is going to get more negative, more negative, more negative. And that's going to drive your overall water potential in the sieve tube element more negative. So then, as that's happening, you'll have a less negative area nearby. It's typically the xylem. And that's what they're showing here, that you have a less negative overall water potential right next door. And so water moves from less negative to more negative. So as those solutes are getting dumped in there, driving the water potential more negative, the water's going to start to flow from the xylem into the phloem. As soon as you get that water coming in, now you get a positive pressure. So your psi sub P value goes up. It's some positive number. So that's describing like the first part of phloem translocation. That's setting the stage for, okay, now we have a nice pressure. But it's a pressure gradient we need to think about. So you have to demonstrate that you understand somewhere else there's a lower pressure. And once you get a pressure gradient like that, then you'll get movement of sugar. So that's what's being shown on this image too. And I use the example of a white potato tuber starting to develop. And so if you imagine sugars needing to get down to that tuber in the roots area, and that tuber is starting to say, hey, you know, I need that sugar, I need that sugar, I need that sugar. So sugar is getting unloaded into the tuber to build a nice potato tuber for your French fries. Okay, so now solutes are coming out of the sieve tube element into the tuber. So now there's less solutes. That means your psi sub S value will get less negative. It'll start approaching 0. And now your overall water potential, because of that, gets less negative. So now water will exit out of the sieve tube element down at the sink over to a more negative area, which is the xylem. So they're showing that you have an overall water potential of negative 0.4 in the sink tissue and to negative 0.6 in the xylem. So less negative to more negative. So now you get that loss of water. Well, loss of water, loss of pressure. Your psi sub P starts to go down. And so that's what they're showing there. Your psi sub P is only 0.3. But up in the source, it's 0.6. So now you've established the gradient idea. Higher pressure in the source, typically the leaves, lower pressure in the sink, in this case with the, in the root area where the tuber is developing. So now you've completed the story. Now you have a pressure gradient and the sugar solution can move from source to sink. And remember, it's all open here. All these sieve plate areas, there's no membranes there. So it's just flowing free from source to sink. They don't have to bother with dealing with any membranes. So really, it's kind of an elegant system. There's not a huge input of energy anywhere. You have just this pressure gradient developing and the laws of water potential, you know, allowing for this gradient to get established. Okay. Let me back to anything else in that slide set you want to know? Some other information in here. Well, we need to move on for mineral nutrition. So knowing a little bit about the soil properties, negative charges on the soil particles, certainly cation exchange, helping with release of minerals off the soil, microbes doing their part. We talked quite a bit about that. And then we got into the pH of the soil system. And then, of course, the action of mycorrhizal fungi. We only covered the two, the VAM fungi and the ecto. So knowing how they interact with the plant, what they do for the plant, what the plant does for the fungus. So the way I would encourage you to study this idea is to look at both ectos and the VAM and kind of do a side-by-side comparison. How are they similar with how they interact with the plant? How are they different? I think that will help you pick out some of the details I'll be interested in seeing if you've looked those over and learned them. So kind of a compare and contrast study method, I think, would be helpful for the mycorrhizal fungi. And then we got into what are the minerals that the plant needs, macro and micro. And the table. So this table, being able to identify where the minerals fit in this table, I guess would be the way to put it. So there's 4 different groups here of what they do for the plant. And then the different minerals that fit into those different groups. So table 4.2 will be a good one. And then mobile versus immobile, make sure that you're good on that. And then the minerals that I have actual PowerPoints on, so nitrogen, phosphorus, and potassium, those are more what you should zoom in on in terms of if there's any detail asked. It would be the ones that I've picked out here and wrote some more stuff up on. And then understanding like when we talk about mobile versus immobile, that's once it's in the plant. That's what I want you to focus on. Like once it's in the plant, and what does it mean if it's mobile or immobile? And then what would the symptoms look like if you start to see a plant that's stressed out because of deficiencies? What would that look like? So those types of ideas. Questions on this slide set? Okay, what about biological nitrogen fixation? So this is a definite process between the plant and its microbial partner. No fungi. No fungi can do this. Only bacteria. So this is a process again, because we walk through the story of, okay, you got this plant and you got some bacteria in the soil, but how do they figure this out? How do they find each other? How do they enter into the relationship? How does the plant kind of control how the bacteria enters, how they set up camp? And so this is a definite process again of, thinking about a plant that needs to form this relationship. How are they going to do it? And how will the bacteria let the plant know, hey, I'm a friend, let me in, let's do this. So it definitely a process. Doesn't come cheap because there needs to be a big input of ATP, but benefits outweigh the costs. So they do it. Questions on this, the process of it? You have the recognition, they have to find each other first before anything else can happen. And then once they all realize everything's okay, they'll start the invasion step. And then the release into the root tissue, and then the setting up camp and the actual fixation. And then we did talk about the enzyme. The enzyme's peculiar. It's a very complex enzyme, but it's got a weakness. It's sensitive to oxygen. So I'd like you to think about that. That's its big downfall. So it creates some problems that they had to evolve and solve that problem. Any questions on biological nitrogen fixation? Yes. Not in full, other than the idea that you have, where'd it go? Nitrogen, dinitrogen going to ammonia and 16 ATP. That's pretty much it. Other questions on this slide set? The light reactions, you're not going to need to know the light reactions in detail. It's just slides up to slide 30 through slide 30. Okay, so the first handful here is just talking about basically light energy is being used. Need to have chlorophyll for that, that there's different chlorophylls. Thinking about slide 12 would be a good one to think about. 11 and 12 in particular, the triplet state. And then slide 12 with blue versus red light. Photoreceptors, you know, the different chlorophyll pigments. Recognizing that there's different types. We tend to focus on chlorophyll A and B in this class because the higher plants have that. Being comfortable with chloroplasts again. Antenna complexes, and then the overall big picture idea of, well, the whole goal is to make NADPH and ATP. But we won't do any of the slides beyond that. It takes a while to learn the details of the step-by-step light reactions. So same idea on Friday. It'll be 2 versions of the exam, multiple choice and then short answer or essay. I haven't made the full, I have a draft, but I haven't made it yet. Right now it's sitting at around 80 points. So I'm guessing it'll be somewhere between 80 and 100. But I'll let you know more on Wednesday. Okay. So if you have questions, you can shoot me an email. You can stop by for office hours as well. Or you can set up office hours if you need to talk to me in more detail. And my office hours aren't working for you this week, we can find another time.